Hello, everybody, and welcome to the second of three webinars in our Season on Spring series. My name is Michael Edmonstone, and I'm the Communications and Engagement Lead here at Shoal. Shoal mobilizes urgent conservation attention for the world's most threatened freshwater fish species, actually all species, but mostly fish at the moment, and through the landmark 1000 Fishes initiative aims to catalyze conservation attention for 1,000 of the planet's most threatened freshwater fishes by 2035. Springs may be small and all too often overlooked, but they are amongst the planet's most extraordinary freshwater ecosystems. They support an outsized share of biodiversity, harboring unique communities and high levels of endemism, while also sustaining river systems and carrying deep cultural and human significance across the world. Last year, springs received an important boost on the global conservation stage. At the IUCN World Conservation Congress in Abu Dhabi, Motion 16, Springs Under Threat, Mobilizing Urgent Action for Neglected Freshwater Systems, was formally adopted. Developed by the IUCN SSC Freshwater Conservation Committee, the motion calls for coordinated international action to better protect springs and subterranean ecosystems, and it has helped elevate these neglected habitats within the conservation agenda. Since then, Shoal and a growing number of partners, including the IUCN SSC Freshwater Conservation Committee, Rewild, the Spring Stewardship Institute, and the University of Missouri St. Louis, in collaboration with St. Louis Zoo, have been working to turn this momentum into meaningful action. This second webinar in our Season on Spring series explores the world of spring biodiversity and the research helping us better understand these remarkable ecosystems. We're delighted to be joined by an excellent panel of speakers who will guide us through the science, the discoveries, and why springs deserve far greater attention. We'll hear from Mary Seddon. Mary is the chair of the IUCN Species Survival Commission Mollusc Specialist Group. She has worked for 20 years as a museum curator, specializing in taxonomy, field surveying, and capacity building for the identification of land and freshwater snails of Europe, Madeira, Morocco, and East Africa. She became an IUCN Red List assessor in 1994 and has been working on red listing since then, so she has seen the evolution of the assessment process from the expert opinion version of 1994 through various iterations and updates to the version of today, which uses quantitative criteria. Mary was part of the working group that revised the categories and criteria between 1996 and 1999, and as a result of that, she holds various volunteer roles within the IUCN Species Survival Commission, including Emeritus Chair of the SSC Mollusc Specialist Group, member of the SSC Invertebrate Conservation Committee, and member of the IUCN Red List Technical Working Group. Mary will talk with us today about the most recent results from what sounds like an extremely intense 3 weeks of workshops across Europe and Australia, which have resulted in nearly 1,000 freshwater mollusk assessments to add to the review. Her talk will relate to the threats to spring snails and the need for conservation actions. Next up, secondly, will be Rajeev Raghavan. Rajeev is a fisheries scientist and aquatic conservation biologist known for his work on the freshwater fishes of the Indian subcontinent, particularly on subterranean and groundwater dwelling fishes. He is an assistant professor at the Kerala University of Fisheries and Ocean Studies and the South Asia Chair of the IUCN's Freshwater Fish Specialist Group. Rajeev is an expert on subterranean and groundwater dwelling fishes and has described 25 new species to science, which includes 5 new genera and 2 new families. These include the world's largest cave fish, the world's only subterranean snakehead, and other morphological novelties. He has authored more than 200 scientific publications, with his research helping to shape conservation priorities for some of the world's most threatened freshwater species, and he has been continuously listed in the Elsevier Scopus Top 2% Scientists of the World since the year 2020. In honor of Rajeev's research contributions to Indian ichthyology, 2 fish species have been named after him, a snakehead from the northern Western Ghats, Chanarara, and a hill stream loach, Indorheanictes regevi. He has been an advisor to Shoal since the initiative's inception in 2019, and Rajeev's presentation is titled Hidden Worlds and Shared Discoveries, Synergizing Public Participation and Science for Understanding India's Subterranean Biodiversity. I said that Rajeev was going to be next up, but it will be Chris, Mary, Rajeev, Michi. No, I've got that wrong. I'll come back to that. Closing off the session will be Chris Martin, an associated professor at the Department of Integrative Biology, UC Berkeley. Chris is the first curator of ichthyology at UC Berkeley in the Department of Integrative Biology and Museum of Vertebrate Zoology. He directs the Fish Speciation Lab, which investigates the rapid evolution of new fish species and their extinction with a focus on extreme environments, from hypersaline Caribbean lakes to Cameroon volcanic craters to the twilight zone of the deep ocean. His lab has also worked with the US Fish and Wildlife Service for 14 years on the conservation of the most endangered fish in the world, the devil's hole pupfish, and he coordinates an annual inventory of captive populations of Cyprinodontidae, including many extinct in the wild species. I'm sure many people here will share my excitement at the title of Chris's talk, Diabolical Survival in Death Valley, and what we can learn from endangered pupfishes. But first, Michi Tobler is going to talk with us about springs as evolutionary laboratories in need of conservation. Michi is a professor of biology at the University of Missouri, St. Louis, and a senior scientist at the St. Louis Zoo. His research focuses on fishes inhabiting groundwater-dependent habitats and on the ecological and evolutionary processes that shape their persistence and diversification. A central emphasis on his work is translating these insights into actionable conservation strategies, and his lab works in close partnership with zoos, conservation organizations, and government agencies to implement science-based approaches to biodiversity conservation. Just a very small, quick piece of housekeeping, and then we'll let Michi take over. Today's webinar will last between an hour and a half and 2 hours. After the presentations, we'll have a chance for a question and answer session. And as always, it would be fantastic if you could introduce yourselves on the chat and say where you're joining us from. Please keep your questions coming in throughout the talks. We'll note them down and ask them in the Q&A. The session is being recorded and we'll upload it to Shoal's YouTube channel and website later today. So Mary, Michi, Chris, Rajeev, it's really fantastic to have you joining us. And Michi, if you're ready to share your screen, it's over to you.
That sounds great. Can you all hear me and see the presentation? Awesome. Thank you, Michael, for this introduction and for your kind words. I'm really excited that we're here talking about springs. And I'm excited there's sort of growing momentum to talk about spring conservation specifically. If you were in the previous webinar, you've probably heard already about the ecological importance of springs, not just for aquatic organisms, but also for terrestrial organisms that visit springs for a variety of stages during their life. And even though springs are, of course, very abundant, they have been largely neglected by conservationists and often by ecologists in general. So I'm really excited about the passing of the IUCN resolution, and I'm excited that springs will hopefully make it towards the top of the conservation agenda in the coming years. Now, I'm a fish biologist, and I think about springs primarily from the perspective of fish. And springs are, of course, critical in terms of sustaining riverine fishes across landscapes. Springs are critical in providing base flows for rivers, especially during times of low precipitation. And they're important thermal refugia for fish during time of environmental stress. But what's really interesting is that fishes, or most fishes, are typically just ecological users of springs. What I mean by that is that fish often move in and out of springs for reproduction or in time of environmental stress. And many fish species that occupy springs are not necessarily spring specialists. So here in Missouri, where I live, for example, there's thousands of mapped springs. Many of these springs harbor endemic invertebrates, endemic plants, but there's only a small handful of fish species that will exclusively inhabit springs. And most of the fish we encounter in the springs around here, they are actually occupying a wide variety of ecological settings, a wide variety of habitats. Now, in other parts of the world, that is very different. There are, in fact, a large number of spring endemics among the fishes. And the situations where springs turn into what I call evolutionary laboratories are situations where springs are very different and represent sort of extreme environment that impose strong selection. Springs that require specific adaptations for species to persist in them. Or alternatively, springs that are extremely isolated on the landscape, where there's very low connectivity for species to move about. And sort of these strong sources of selection, isolation from other aquatic habitats, and long-term stability that promote divergence among fishes and ultimately create spring endemic species that are adapted to very specific environmental conditions and that are microendemics, often to a single spring or a few spring complexes. So it's sort of these microendemics that I want to talk about today. And I hope what you take away sort of from my talk is that environmental extremes are what can generate evolutionary novelty. And in conjunction, along with it, they create conservation vulnerability at the same time. So I'm going to talk about sort of two examples of spring fishes that epitomize sort of this isolation in sort of weird spring habitats. And that is first hydrogen sulfide rich springs. And these are toxic springs dotted across landscapes around the world. But we've mostly studied them here in the Americas. And I also want to talk a little bit about desert springs, also primarily here in North America. So let's talk about sulfide springs. What are sulfide springs? Well, sulfide springs are springs that are fed by groundwater that has been enriched underground in toxic hydrogen sulfide. So these are natural processes usually associated with volcanism or hydrocarbon deposits that sort of enrich this toxic gas in the water. And as the water gets discharged to springs, they create this toxic habitat that is a sort of fairly unique and ecologically different from adjacent non-sulfidic habitats. Hydrogen sulfide is toxic because it shuts down the mitochondria in organism cells. So it basically prevents cells from producing ATP aerobically. Organisms run out of energy, and as a consequence, they die. And just to sort of illustrate how bad this really is, in most of the springs we study, we find hydrogen sulfide concentrations that are tens, hundreds, and in some cases thousands of times higher than what's considered lethal for most aquatic organisms. And if you put regular fish that are occurring in non-sulfidic habitats into these waters, they typically die in a matter of minutes to a few hours. So these are extremely toxic conditions, unfavorable for most animal life. At the same time, these springs are extremely energy rich because hydrogen sulfide fuels bacterial primary production, similarly like hydrogen sulfide fuels primary production in deep sea hydrothermal vents. So these springs are occupied by sulfide oxidizing bacteria that use energy from the sulfide, just like plants use energy from the sun to produce biomass. And it's this bacterial biomass that's essentially the base of the food web for these sulfide spring ecosystem. So if you can make it in a sulfide spring, there's a lot of advantages in being in there, in that there's a lot of resources to be exploited. Now, many fish species have colonized sulfide springs. We know most about fishes of the family Pisciidae. Those are the life bearers that have colonized sulfide springs in the Dominican Republic, in Florida, and Costa Rica. And then there's a huge diversity of sulfide springs and sulfide spring fishes in southern Mexico, particularly in the states of Tabasco and Chiapas. And this region in Mexico is sort of the primary focus for a lot of our work over the past 20 years. I want to emphasize that these springs are not large. In fact, most of these springs are relatively tiny. This is one of my favorite springs, La Esperanza spring in Chiapas. And you can see the spring head is located right below this tree here. And the spring flows for about 100 to 120 meters before it merges with the Ixtapangahoya River. This is the same spring, just seen from the ground. Again, there's this large tree, a remnant of the original forest. The spring heads are located up here. Water flows, and it essentially merges with the Puya with the Ixtapangahoya River. And there's really no physical barriers that would prevent fish from swimming in and out of the spring. However, if we go sample fish, life bearers, outside of the toxic water and then inside of the toxic water in a matter of maybe 20 or 30 meters, and we conduct genetic analysis on these fish, we find over and over and over again that the fish in sulfide springs are starkly genetically different than their ancestors in adjacent non-sulfidic waters. So despite of the lack of physical barriers, we see strong genetic differentiation. And that's because survival in sulfide springs requires physiological adaptations. And in fact, we see convergent evolution across a lot of these lineages. Convergent evolution in physiological pathways that help the fish break down hydrogen sulfide to detoxify it. This is this SQR pathway. It basically oxidizes sulfide to non-toxic forms that can be excreted. And we also see modifications to the toxicity target in the mitochondrial respiratory chain. So when the toxicant is present, the mitochondria of these fish continue producing ATP. And it's these physiological survival mechanisms that are required for survival in these toxic springs. Sulfide fish have also diverged from their ancestors in non-toxic waters in a wide variety of other traits, ranging from respiratory structures and oxygen transport proteins. They have modification to their jaws and their guts that allow them to exploit their bacterial resources. There's changes to their sensory systems and their brains, to their pigmentation, to their reproductive life history. And so, as a consequence, we see these phenotypically very divergent forms in sulfide springs that are genetically distinct and genetically isolated from their ancestors in non-toxic habitats that are just a matter of meters away. Now, you can imagine the simple small size of these habitats render these populations and species vulnerable. And in fact, there's only one sulfide spring that I know across all of the ones that I have visited over the past 20 or 25 years that I would consider relatively pristine. This is the El Azufre spring near the village of Tapijolapa in Mexico that has a relatively intact riparian buffer and only moderate human impacts through tourism. Most of these springs are in fact very heavily impacted by human activities as they are embedded in sort of an agricultural landscape that is dominated by human activities. This is a spring that exemplifies this. This is the Baños del Azufre spring in southern Mexico. You can see the spring heads are primarily located here. We have a long spring run. This is one of the larger springs that stretches over about a mile of toxic habitat. When I first started working here, the landscape looked like this area here in the center. Lots of trees, small pastures that were used for subsistence farming. But in the past 20 years, we've seen largely clear cutting for cattle pasturing. We've seen planting of oil palms. This is what you see in these highly sort of dotted symmetrical looking plots over here. These are all oil plant plantings that now dominate the riparian areas of the sulfide springs. The actual spring head is now located in a hotel complex. Most spring heads discharge into swimming pools that are used by humans. And to make matters worse, the sewage treatment plants of the community here upstream has been broken for several years, and we have discharge of raw human sewage that flows into this spring complex. So to make a long story short, the vast majority of these springs are heavily impacted through habitat loss and pollution. and the impacts on the fish are significant. So what we see frequently happening, especially here at the Banos, are actually massive fish kills that come in conjunction of swimming pool maintenance. And what you see here is a video where literally hundreds of thousands of fish were killed in a matter of an hour or two. And these are all individuals of two endemic species, Picilia sulphuraria and Gambusia eurystoma, that occur nowhere else in the world. So the situation is dire, and these sulfide spring lineages are in desperate need of conservation action. And so far, none of the species or population are receiving any sort of meaningful attention. Now, the other group of fishes I think about a lot are fish in deserts. These are fish that occur in sort of the arid landscapes of North America. And you can see the sort of the extent of deserts here highlighted in this orange coloration. Much of the western United States and north central Mexico are considered deserts. And there's about 200 species that exclusively inhabit these sort of desert habitats, many of which are spring dependent because they exclusively inhabit springs, or they're dependent on the flows generated by springs in these desert landscapes. There's about 200 species in this area, as I mentioned. Most of them are in the order Cypriniformis and Cyprinodontiformis. Together, these two orders make up about 60 to 70% of the fish fauna in this region. And you'll hear a little bit more about the pupfishes that Chris will talk about later in this webinar. Desert springs are a really weird habitat. Most of them are tiny. What you see here in this picture is Sago Spring in New Mexico. It's home to a population of Dionda episcopa, a small minnow. And the combined habitat of this minnow population is maybe 3 or 4 parking spots. So I'm always amazed how some of these populations can persist in tiny habitats. And they're extremely isolated from other populations. And they're subject to a lot of environmental stochasticity. So spring flows might vary through times. Temperature might vary through times. And so these fish populations are not only exposed to strong sources of selection, but also lots of genetic drift because the small populations fluctuate through time in a random manner. Whenever we have strong selection and strong genetic drift, in conjunction with sort of the geographic isolation in these desert habitats, because springs are not connected through surface waterways, we tend to get divergence. And the consequence is that in these desert springs, we find complexes of species that are ecologically very similar, but they have been isolated for extended periods of time evolutionarily. And what you see here is just one example of this. These are species of the Gambusia nobilis complex that dot springs along the Pecos River in the United States, in Texas and New Mexico in the United States. Now, all of these desert species, they're in bad shape. A graduate student of mine, Casey Ernest, has recently done some extensive analyses comparing threat status across non-desert fishes and desert fishes, both those that occupy springs exclusively and those that do not. And what you can hopefully see here is desert spring fishes particularly are disproportionately threatened by extinction. And this is they face about a 3 times higher extinction risk than your average non-desert fish in North America. And the key issues here are really the disappearance of habitat because of water abstraction, both abstraction of surface and groundwater, because of land use changes, and that destroys habitat, and also because of invasive species. And the big question, of course, is what are we going to do about this? What are we going to do to prevent many of these species from disappearing for good? And so we spend a lot of time in my lab thinking about how can we promote conservation of microendemic extremophiles. We're trying to establish meaningful baselines for assessing demographic and genetic population health. We've been spending lots of efforts, mostly using genetic tools to evaluate current conservation practices, trying to figure out what works and what doesn't. And we're trying to figure out how we can develop adaptive frameworks for population management in support of existing populations, but also in attempts to reestablish populations where they have disappeared over recent times. An important test case for us is the Pecos mosquito fish, Gambusia echeliorum, that occurs around Bitter Lake in New Mexico. This species occurs only in a handful of springs and spring-fed sinkholes on Bitter Lake National Wildlife Refuge. And it has disappeared from many of the locations it historically occurred. We now know of 9 populations. Only 3 of them are deemed relatively unimpacted. And we're at the same time, there's a wide variety of sinkholes that provide suitable habitat that might be suitable for translocations. So a lot of our work is trying to figure out how do we best conduct these translocations, and how are we doing so in being cognizant about the fact there's 5 other threatened and endangered species on the property. So how do we manage one species without having detrimental effects on other species? Ultimately, the goal is not just to do good and do right by the Pecos mosquito fish, but we want to use tools that can be applied widely. We cannot tackle these conservation challenges on a species-by-species basis because there's dozens of species in urgent need of help. So a lot of time we spend thinking about how we best integrate insights across species and develop evidence-based conservation strategies that stakeholders can pick up and apply in their own conservation context. I think a critical part of this is building more capacity for ex situ conservation in freshwater fishes. And this is another resolution that passed in the last World Conservation Congress, along with the Springs resolution. There is a concerted effort of motivating zoos and other stakeholders to invest more in establishing ex situ populations for threatened fishes. And we're in urgent need of establishing good practices that allow us to manage these fish in a manner that they can be returned into the landscape in due time. And so with this, I want to thank a bunch of collaborators and funders. I have not listed all of my students here who are doing a lot of the work. I thank you for giving me the opportunity to speak, and I'm looking forward to addressing your questions later if anyone has any. Thank you.
Thanks so much, Michi. That's fascinating, and some really beautiful ecosystems there, despite the high levels of hydrogen sulfide. And amazing that the species thrive in such toxic conditions. Mary, if you're ready to pull up your presentation, it'd be fantastic to hear from you next.
Just trying to get back in there now.
Brilliant.
Am I in the right place now?
Yeah, it looks great. Thank you.
Okay, thank you. Okay, so there will be a little bit of an overlap, fortunately, in a way, with Michi's talk, because I'm also going to talk about one of the species, but only very briefly at the very end. Mine is a little bit more based on the results of some of the data that we've been gathering as part of the Red List process. But then looking through to why freshwater springs in particular matter in a context of molluscs. And behind me, the scene that you've got is one particular type of spring, the mound springs in Australia. But the data that I'm probably going to look at most in this particular presentation is the data that we've just got from Europe, where we have actually reassessed 800 species that we last assessed in 2011, which allows us to look at changes. And I'm also going to look at some of the overall data sets and how that compares to some of them, and how you can use the red list data and the classification schemes to extract your own information if you're looking at wanting to answer questions. So I have to say that it is not, I am just basically presenting this data. There's been at least 300 people involved in this project overall. So bear with me if I don't know the details of every small species that are in the system. So I'm going to start off with looking particularly at the Mediterranean region. Then I'm going to talk a little bit about how that goes over to the Convention of Biological Diversity targets for 2030, 2050, and how we might look at the data in the context of the red list data. And then look at why freshwater springs in particular are habitats at extreme risk. So the CBD targets, you'll hear CBD quite often referred to when you're looking at governmental reports. And every 10 years, those targets change. And they do an overview of how the targets were back at the beginning of the 10-year, and then they change and amend those targets. These targets were originally the HE targets, which were in place until 2020. COVID sort of interrupted the review process of that. So the new targets weren't agreed until December 2022. And those targets also overlap and align to the United Nations Development Programme's Millennium Goals, which came into effect and started back in the year 2000. Now, the important thing for anybody to note is that if your country or state is a signatory to the CBD, they should have a state agency that is responsible for monitoring progress on implementing plans for biodiversity. So in theory, probably in the case of the States, it would be the US Fish and Wildlife Service. In the UK, it's JNCC, which is the Joint Nature Conservation Council. But what you will find is that quite often they only report on the larger species, for example, the birds and mammals. And very rarely do they start looking at other systems, and particularly not the freshwater systems as such. So it's worthwhile starting to think about this and maybe ask questions within your own country. Now, why is the red list data important? Well, the IUCN Global Red List is one of the tools that they use for monitoring against the CBD. The database is updated on a twice a yearly basis, typically. Next year, this year it might be slightly different because we're in a changeover phase and the system is being developed. But any submission to the red list is treated rather like a journal. It basically does go through a peer review process. It goes through a technical scrutiny process to check that it meets the categories and criteria and has been consistently applied. And from the point of view of molluscs, at the moment, we've got about 1,200 draft assessments from Europe, Australia, Indonesia, and Russia in particular, because we've got a big project running to try and complete the global freshwater mollusk assessment. Any species that is published on the red list gets a DOI number and a PDF file, so that there is always a way of tracking what the species assessment was at any point in time, because assessments are changed, particularly birds. Birds are updated every 5 years. The molluscs now, we have got something like well over 1,200 species that will have been assessed more than once. So you then start to get an archive of old assessments. So in the case of molluscs, what sort of data is required? Well, for the widespread species, which are basically the minority of species, we'd be looking at estimates of population decline based on generation time. The problem with that is that quite often we don't have big population data. We're invertebrate species, and often that sort of data isn't gathered. So we end up having to use proxy data for that. But in general, the widespread species are not the ones that really tend to be much threatened. I'll caveat that because I'll come back in a minute to where that might not apply. Typically, the reason that we've got the red range restricted species, it's most of the mollusk assessments will be based on the number of locations, the general distribution, the number of sites that they found out, the quality of habitat there, whether sites have disappeared. And most of the spring snails that we'll be talking about, and spring snails are a very hyperdiverse group within mollusks. Most of those basically are range restricted, typically less than 5 locations. By a location, I mean not necessarily a site, but an area where the groundwater source is feeding all little springs within a small area. So the information that we come to assessment workshops with, or that the assessor looks at, is typically then filled into the database. And we will also be capturing other important information like, is the species particularly tolerant of pollution? There are some that are tolerant of pollution, and therefore pollution as a threat isn't something that's a big threat to it unless it's really catastrophic level of pollutions. You get species that are quite tolerant of disturbance. Again, so they would tolerate cattle movement around. Other species are very sensitive to that. Other species require really pristine water conditions. So that sort of information is the information that we are capturing. And if you're trying to use climate change projections, typically you're supposed to be using a particular data model. We won't look at that at this moment because at this moment in time, we haven't actually started to use climate change projections because the time scale compared to the generation time of molluscs is not relevant, but it might be relevant to other groups. So if I am looking at the overall data sets of the last update, and bear in mind there will be a lot of molluscs in the next update, 2026.1, which will be coming out fairly soon, hopefully in July, you will see that on the left-hand side here, you have all species that are currently on the red list and their typical division of critically endangered, vulnerable, near threatened, and the least concerned species. And with all species on the red list, the circa 52% that are on the red list are not considered threatened at all. By contrast, when you start to look at all of the global molluscs, now this is including both the marine molluscs and the land snails as well, and when I'm looking at this particular data set, you will see that there's a much higher percentage of extinctions, and there's a lower percentage of species that are considered to be least concern. But that in general, why we're focusing at the moment is we are about 20% off as being effectively an almost complete assessment. If we look at how we are tracking change, in general, the threatened species of freshwater mollusk, now I'm now just looking at the 4,000 odd species that were in the 2025.2 update. And of those, for freshwater alone, we are talking between 32 and 39% are threatened with extinction, and a much lower percentage, 30%, that are least concern. If we were to add in the species that are considered critically endangered and possibly extinct, quite a lot of these are freshwater species that are found in single sites where the habitats have changed, but we suspect there might be a possibility that they are hanging on somewhere in a small little bit of habitat that we've not been able to get to. In general, again, you're seeing that between 2.6 and 4.2% are actually already extinct. By contrast, only 30% are considered least concern. But that in general, if you go back each time we've actually updated, you'll see that quite a similar percentage end up in the different threat categories. Now, when we come to looking at springs on the red list, and this is a paper that's in submission at the moment, but isn't accepted, so please don't take this particular diagram as it's an indication of research in progress from Joseph Gregor. And he was pointing out how difficult it is to classify springs in any way that's particularly useful. And on the red list, all we have is effectively surface water spring situations and underground spring situations. But that in reality, life is a lot more complicated, and it depends a lot on where the water source is coming from, where the species is living within that complex system, whether it's within the fissures, whether or not it's interstitially, whether or not it's in the cave itself, whether it's a spring that's on the slopes or basically a groundwater fed pool. But if we were to basically take off you can do this within the red list data. If you were just to take the freshwater molluscs within springs in its broader sense, you will find that basically only 20% approximately are considered least concern, and nearly 46% are considered to be threatened with extinction. And there's a fair number of species that are sitting as near threatened, and this is where they're quite often species that are at less than 5 sites, but where there actually isn't any threat that is prevailing at this moment in time, but that longer-term climate change might impact them such that they will rapidly go into one of these higher categories. We still have a percentage of species that basically are uncertain, and that is because we either haven't recorded them living, they're only known from dead shell material, and it's uncertain where within the system they come from, whether they're a surface water species or whether they're an underground species. But in general here, you can see that for the spring snails, drought is now considered to be one of the major impactors that's causing the threats, with a secondary level at the abstraction of groundwater. Now, I'll come back to this in a minute, and that pesticides are certainly also potentially impacting 20% of those species. And that's 20% of all species, not just the threatened species. So when I said that we were able to look at change, we've been able to look at change because if we look at this data set here, this is for the Mediterranean region based on the data that was gathered between 2011 and 2014. And here you'll see that you're seeing a very similar pattern, large percentages of species that are considered actually threatened, and a very small percentage of species that are actually considered to be least concerned. Now, bear in mind, you're now in the Mediterranean region where there are very high pressures from touristic use and agricultural use. And most of the freshwater sources there are exploited for one purpose or another. In addition, there are also poor controls on both pollutants from sewage and from nutrient loads. Eutrophication is quite a big thing in quite a lot of the Mediterranean water systems. A secondary also pollution is sedimentation, and that is usually where you're in an area that gets a lot of burn, and therefore when you get any small storms afterwards, a lot of sediment is washed into the system because there's no longer any vegetation holding the sediment in place. And this is what I was meaning about seeing a change. If we look at the new data set, and it's not quite, the data hasn't quite been finalized, so I couldn't show you a little graph. But in general, the new revised versions that will be coming out in the next Red List updates within the next 2 months will show that increased frequency of droughts and the abstraction of groundwater and surface water are now the two dominating threats, whereas droughts originally was down at a much lower level, I think it was the 3rd or the 4th level of threat. Now that is a change that has happened within the space of 15 years. And it's reflecting the fact that in part we are seeing change in seasonality to the rainfall patterns. The rainfall, which traditionally would have recharged the groundwater systems in wintertime, hasn't happened for several years. So we have changing patterns of rainfall when it comes, and also increased drawdown and increased frequency and intensity of drought events in the summer periods. So such that we're actually seeing habitat conversion. So how does this data get used, or how could it be used more effectively to influence policy? Part of what the red list data could be used for more effectively within, for example, a European context, is to use it for environmental impact assessments at major sites. Dam development in certain parts of Eastern Europe has created quite a lot of change for groundwater systems there. But because snails don't count, they're not on any legislative requirements, they tend to be overlooked when any environmental impact assessment is done. So by putting this information into the red list, it should make that information more accessible. It should be easier to find the information when an environmental impact assessment is ongoing. We can use it to basically identify areas and habitats that need action. We could, in theory, use existing opportunities for incorporating actions for habitats. And within Europe, there were two big projects looking at whether or not KBAs could be identified, KBAs being key biodiversity areas, for freshwater systems. We identified quite a lot of species that actually, in theory, should have triggered the placement of a KBA area. However, the guidelines that are written to how these get implemented mean that you basically need population data to support the fact that the species is there and is in decline. And yet for invertebrate species, this data is rarely available. So the KBA guidelines need a little bit of a rethink if we're actually going to implement them effectively for freshwater invertebrates. And we already know that in many parts of Europe, for example, there is no requirement to increase to meet the 30% of land protected by 2030, because in many areas, they've already done that by just using the important bird areas and other equivalent Ramsar sites and special areas of conservation, the EU designated areas for habitats directive species. And there are very few molluscs that are basically in those habitats directive annexes. So it's limiting at the moment to actually incorporate action for habitats, and yet that is probably going to be the most effective way of actually conserving some of these species, especially small range restricted species like freshwater spring snails. The other thing is also to basically get, there are national parks that are in protected areas, making certain that that is documented adequately. And one of the things that I wanted to sort of just show a little bit is how complicated it can be. And I think this was touched on in the last series as well, Michael, when I think both Larry and Heidi were talking about the complication of overlying catchments and underlying groundwaters. And here you can see the number of species that might be impacted within a groundwater system where the inflow to the system is over here with a range of species that are so surface, underground, but there is movement across quite a large area. And then the actual outflow points are up here to the sea. This is actually an estuarine area with a coastal spring here. And yet you have a range, all of these species here are range-restricted endemics that are mainly threatened with extinction. Some of them are threatened at vulnerable, some of them are more critically endangered, depending on the level at which they are. And they're underground, and this here is a concrete, I think, barrier that was put into place as part of the spring management, because the spring here provides most of the water for the two nearby towns. So the exploitation is going on here. That's the spring here. And that is the power station that is also using the spring to generate HEP, hydroelectric power. And the main river channel, you can see, is quite heavily canalized. So that's the river that's coming in, that's part of the source over there. So catchment management in this situation would be, in theory, the way that you go about doing it. But as already alluded, the Water Framework Directive, which in theory should conserve both the groundwater biodiversity and the surface biodiversity, probably doesn't include most of those species because of the way that it was written. So we are able to map red list threats against the CBD targets, and this is available in Bolam et al. 2022. They looked at, these are the categorizations in the red list data, and this is the target within the CBD that the species, these numbers here reflect the number of species impacted. This was in 2022, so that this would have been the 2021 data set that was being used. And these are the targets that are within the CBD as written. And these are probably the main species areas that we are concerned about managing for a freshwater mollusk perspective. So just briefly to touch on just a few points, if we were to look at target 2 and habitat restoration, which requires 30% of habitats to be in restoration programs, changing natural system modifications by 2030. There's a lot of work, for example, on the removal of dams to enable free flow. However, there are quite a lot of range-restricted threatened species that live in the marginal riparian habitats that might be impacted by those works. So if you know of projects like that are ongoing, what you might want to check is whether or not actually any implementation work to remove dams has taken into effect what might happen to the riparian habitats that host some species of mollusk and other small invertebrate species, and probably fish as well. The restoration of degraded mound-type springs after borehole capping has been done, but it's invasive species control that's now needed to restore, and actually restoration of the links between the pools in order to move forward. There's a lot of upland moorland restoration projects going on across Europe as part of the trying to reduce flood events. And again, the likelihood, if they was of actually taking into account range restricted threatened species, they might not think about things like that when they're moving forward. Target 4 is very useful because it basically allows potentially species management actions. It requires governments to report on recovery actions for species. But we haven't actually got many freshwater molluscs actually listed as requiring species recovery plans other than for the freshwater mussels. And I give an example here. The only example I can quote for Europe where we've tried that, it failed. So it was quite heartening to see that Percolopsis roswellensis, which was also referred to by Michi, there's been some translocation of about 15,000 snails across the two different species. And initial reports were quite successful. I would like to see reports a few years on, because within Europe, when this was tried, it was initially quite successful for the first 2 years, but 5 years down the line, drought had taken its impact. And therefore, the only example there was not very successful. And the other big one would be to target 7 pollution to control excessive nutrients by at least 50% by 2030, reduce pesticide reduction by 63%, and eliminate plastic waste. And you'll see there's large numbers of freshwater mollusks that would benefit from those actions in theory. But how one goes about monitoring that and how one looks at the after effects, the questions that were coming out of last session and I think also apply here as to how we move forward in terms of monitoring. And finally, to look at managing invasive species. Sorry, Michael, I'm aware I'm probably now overrun. Got a couple more slides. But it's worthwhile saying here that invasive species wasn't a major problem back in 2011. And now, in 2022, when the bivalves were looked at, every single freshwater mussel in Europe had been impacted by an invasive species. Again, over the space of 15 years, major changes. And this is something that we can expect to see more of. So the next steps for us are to basically, once we've got the 2026.1 update, we'll be over at least 80%, hopefully more closer towards 90%. We need to increase and strengthen conservation actions, and there will be a summary paper, which will add to the original paper by Sayer et al. That looked at the impacts of freshwater on fish, amphibians, crayfish and dragonflies. This will change that picture. And we need IUCN to establish that task force on springs as mandated in the motion, look at novel ways to monitor habitats, how we can work with other agencies to monitor recharge and depletion of groundwaters supplying the springs, and look at habitat restoration to see whether or not species can be translocated as an ex situ action. I wasn't certain about that until we've been talking in Australia and North America about the very success of that there. I think all I have to do is say thank you to the over 300 people that have been involved in this process, to Arisha, who's listening in, for all of her help in the 2 last workshops and the upcoming 3 workshops that we have going on, and to other people that are online that have been involved in ex situ breeding, and others that are just starting off with a small MBZ grant, trying to do some conservation actions in Indonesia. And if you want to actually see more about this, try looking at Tentacle, the newsletter, which tries to sort of reflect what we're doing across the world. I think that's back to you then, Michael.
Thanks. So
thanks to Joel's and everybody here for listening in and letting me contribute this.
Brilliant. That was a really interesting insight into how how molluscs are affected by sort of fluctuations in springs and the sort of red list process as well. Thanks very much for that. I actually have a question from that. We're doing okay for time. I have a very quick question. You mentioned that there have been these changing threats due to decreased rainfall and more drought. Has that been over the sort of past 10 years, 20 years, further than that? And also on that, are the threats accelerating? And is there anything that can be done to mitigate them to sort of safeguard the biodiversity?
Well, number one, it depends on where you are in the world as to what data we have to actually look at that. The data for the Mediterranean region, we have the advantage of Copernicus, which some of you may be aware of, which actually does a lot of drought monitoring across. And that there have been certain years, I think it's about I think 2014 was a major year, 2019 and 2022. So we are seeing basically increased changes. And that data is all available on Copernicus, so you can actually go back and rewind on that. The same, I understand, is true for Australia, but I'm not certain whether they were the same years. But in the workshop in Australia, there was major event in 2019 to 2022, plus, of course, all the fire incidents there, which also had different sort of impact. So, yeah, I think in terms of, I do get the feeling that it's a combination of threats coming together, though. So we're already got too much drawdown. We're already seeing that basically increased intensification of agriculture is happening alongside that. So it's not just one threat factor, it's a combination of threat factors all coming in. And it's more so within the Mediterranean zone itself, as opposed to further north. Some of the talks that you had last time were based on the Alpine situation. There it's different. There it's more that more of the problems are related to changes in rainfall patterns and not having the winter snowfall in the same level 3 years on the trot. If you don't have the winter snowfall, you don't get the recharge and the seepage through. So it's, as far as I can see, it's quite complicated. Any answers to that? Trying to control inappropriate management, because people's answers to it is to go and concrete the springs. So you do that and then you destroy all the habitat around it. And that's been a problem, I know, in North America as well. I was reading a paper about all the different types of ways that you could actually cap a spring or make a tank on it. And the same is true in the other big spring snail area in Australia. That doesn't mean to say there are not springs elsewhere, but it's where a lot of the endemism is that's been recognised currently. I'm not certain whether that covers all the points you asked.
It just sounds like an incredibly complex area, yeah, it would be good. Thanks for signposting to Copernicus. It would be good to know sort of maybe it's one for the whole panel at the end, but yeah, what more can be sort of done to safeguard the biodiversity and changing springs? But I know that's opening a Pandora's box of quite difficult conversation.
But I think that's the advantage of actually having that motion at the high level within IUCN, is that's bringing the issue to a completely new set of people that may not have been aware. And it's our opportunity now to basically maintain lots of small, snappy stories is really how you're going to keep it in the public. Because a lot of it is also, a local action. Because in many of these cases, it's a local community. We spent a long time talking to a Moroccan agricultural consortium about basically maintaining irrigation channels that were arcs for species. So they didn't need to worry about using that one for the water, but basically don't do any cleaning on that one. That's the one for the snails.
Thanks so much, Mary. And so from Europe and Australia to India, Rajeev, are you ready to present about the subterranean species of India?
Is that all right? Is the screen visible? Yeah.
That's great. Thank you.
Thanks, Michael, for this opportunity to contribute to this webinar. And it's always a pleasure working with Shoal, often contributing to these exciting events and so on. What I'm going to do today is to take you through tropical groundwater ecosystems in India and highlight some of the work that we've been doing with support from Shoal, and also introduce you to some of the world's most bizarre groundwater fish species that live in these systems. So yes, we know that around 300 species of freshwater fish live in really inaccessible waters beneath our feet. So these groundwater systems, caves, springs, all of that. It's poorly documented, one of the most overlooked freshwater fish faunas around the planet. Out of this, around 90% of them live in caves, which comparatively are the more popular ones, with regard to subterranean fish. But there's also these obscure fish that live in groundwater aquifers. So 10% of these 300 odd species live in groundwater aquifers around the world. So my talk will be more or less focused on these groundwater dwelling fish. So if you look at subterranean fish in general, it's found all over the world, with the greatest diversity in China, parts of Southeast Asia, Mexico, Brazil, the US, and of course India is one of the hotspots for subterranean and groundwater fish. Yeah, so this is what we have. So we, are also home to the world's largest cavefish, a fish that we contributed to the description a few years ago, and lots of other interesting fish that I will take you through in the next, 15, 20 minutes. So when I started working on freshwater fish in general 15, 20 years ago, there were these very rare, obscure freshwater fish that were only known through these very old historic line drawings or illustrations, as we call it. So nothing else was known. There were no photographs, no videos. So they were all known just from one specimen, mostly the type on which these fish were described from. So this was when we started to work on these fish, trying to understand more about this fish. These groups of fish. Yeah. So the main question before us was, where do these fish live, and where do these fish appear? So we talked to people who had knowledge about this fish, the people who described this fish, say 50, 60, 70 years ago. There were very few people who had known this fish. But from looking at the different source of information that was out there, we knew that the fish appeared in really interesting systems. One of these systems were dugout wells. So these are wells which are used for water for household use throughout the country. So these fish appear in these shallow and deep wells. They also appear in these bore wells, for example. So when you pump water from these bore wells, there are these interesting fish that come out. So this is another system in which you find these interesting groundwater fish. These wetland ecotones, where you see a new well being dug up just next to these wetland, and also there are these shallow wetland areas. So there are these fish that are found mostly when a new well is being dug up, or a well is being cleaned, for example, and an old well being cleaned. So you get sometimes you find fish near these wetlands. Of course, there are also fossorial fish that are found throughout the wetlands. They come up during certain times of the day. In our experience, it's mostly midnight, between 12 and 4 a.m. in the morning. So that's when you find these fish foraging in these wetland systems. You find them also in these agricultural plantations, where you have water channels for irrigation between these plantations. So this is a banana plantation where you have these water channels between them. So sometimes you also find these interesting fish out in these shallow channels. Backyard garden pools like this one is also a favorite place for these fish. Sometimes they also appear in these systems. Of course, caves, as I said, the most popular subterranean habitat, which is been studied throughout the world. So we also have caves, but not really long caves, but smaller cave systems in the southern part of India. So the next question before us was, how do we find these fish that are out of sight? So these live in very inaccessible habitats. So how do you get hold of this fish? How do you get hold of fish that are found in groundwater aquifers? So yeah, so this was our answer. The fish needs to come to us rather than we going and looking for the fish. So we need to be extremely lucky. For the last 10 years, that is what we have learned working on these fish. You need to be extremely lucky to find the fish. So we normally wait for the fish to appear rather than go and chase the fish, like we do in rivers, lakes, ponds, wetlands, and other surface water habitats where we otherwise work. The other possible source in front of us was to find the right people who knew the fish, because, as I said, there were lots of old scientists who described this fish 50, 60 years ago. There were also people who collected this fish opportunistically, accidentally, when they came up in their wells, in wetlands near their paddy fields, etc. Most often what people do is to put photographs and videos out on the social media, not necessarily identifying them as fish, but as strange animals, strange creatures that came through their water distribution systems. So there were lots of social media photographs and videos. There are also a particular community of people, a skilled expert community that are involved in digging up of new wells in southern part of India and also cleaning up wells. So we have this habit of cleaning up, draining the entire water from the wells in the summer just before the new rains come in, and that's also done by this particular community of people. So we also started to work with them because we knew that they are one of the most important source of information, also just not information, but also source of fish, because they tend to find these fish when they dig up new wells or clean the wells during summer. So to find the fish, we knew that we need to find the right people. So we started to mine a lot of social media information. There were lots of stuff there on social media, mostly on Facebook, on Instagram, and all that, where people had collected these fish, but not knowing that they were fish. They were called as worms and juvenile snakes, etc. Photos were put up, so we built up a database of locations where we knew the fish had occurred in the recent past. So that was also a way to get to the right people who knew about the fish. So we knew that public engagement is one of the most important or the most important approach to take. So working together with Shoal, the project kindly supported by Shoal, we started to work on a citizen science network, lots of public engagement programs, outreach programs with school kids, with women in the villages. And over the last 10 years, we built a big network of more than 700, 800 people around the region who are now informants, providing us with the source of locations where these fish occur, photographs, videos, any kind of information that was otherwise just not available to us as scientists. As I said, it's just impossible to go and look for these fish in these very inaccessible systems. So local communities, people are the key. Citizen science is probably the only way in which you can improve our understanding of these interesting fish. So that's what we've been doing for the last 6-7 years. So citizen science as an approach for biodiversity documentation was extremely strengthened in the region as part of our work with this fish. With this group of fish. So our team routinely visits almost all villages where you have these old wells, talking to people, showing them photographs, videos, telling them that there are these interesting fish that you can find in these wells, and also to inform us when they come across any of these fish. There were also daring students from our team who went down these wells when the water was a little bit lower during the summer. You can also see them in small caves and crevices, all looking for these interesting tiny fish. The other photo that you see is when a new well is being dug up. You have water being pumped out, to clean the wells, etc. So we normally put a fine meshed net, hoping to find some of these fish that could escape through the water from these wells. So it's a combination of approaches that we take, public engagement, citizen science networks, also dedicated field surveys. So, and we often also see these media stories of these fish slipping out of water taps, water distribution systems. You find fish in your wash basins, for example, on your washroom floors. They're everywhere. So this was a brief introduction on the habitats where they're found, the approach that is required to understand this fish. Now I'm going to quickly take you through some of these interesting fish that are found in these groundwater and subterranean habitats. We have eels, fossorial eels of the genus Ophiuchthys. So 3 species of these eels, very cute little eels that are often confused as juvenile snakes, worms, etc. by the villagers, but they're all fish, obviously. There are also relatives of these eels which are blind. So these are blind aquifer-dwelling eels of an endemic genus known as Rectomictus, again from the same family, Finbranchidae. The 5 species of these blind eels are distributed throughout this region. You have catfishes, for example. So this is a fossorial catfish, Cryptoglannidae, a monotypic species, just one species in the genus and family. So this is an endemic family of catfish to the southern Indian region. They're just a very beautiful fish. So you can see that they're bizarre in many ways. So this one is blind, obviously. This one has no dorsal fin. There's no fin on its dorsal side. You can see the spectacular anal and caudal fin fused together into a fan-like shape. So they all come in bizarre morphological characters. This one is a cute little blind aquifer-dwelling catfish of the genus Horoglannis, a fish which we know very little of, and even aspects of its systematics, for example. So we don't really know its familial position and its relationships with other catfish. So that's a work in progress. We have 4 species of these cute little catfish that live in these aquifers, blind fish. This one is another interesting fish. It's a fossorial snakehead-like fish of again, a monotypic family, Enigmachanidae, just one genus and species, endemic to this part of the world. A spectacular fish which does not have a pelvic fin. So this is another morphological novelty. This one is the most recent fish that we have described from the northeast part of India a few months ago in February, March of this year. It's an aquifer-dwelling loach, a new genus of the family Cubitidae. You can see the small eggs, tiny eggs in this fish. There are also aquifer-dwelling eel loaches, for example, of the genus Pangeo, which are otherwise a common a species that a common genus that is found across South and Southeast Asia. But in southern India, we have 2 species of eel loaches that are completely subterranean in their habit. So these are aquifer-dwelling eel loaches. This one is the world's largest cavefish, which we described 3 years ago from the northeast part of India. So this lives in caves, unlike all the other fish that I just showed you, which are all aquifer dwelling or fossorial and live in wetlands. So these are true typical cave dwelling fish. So it's just not fish. There's also an shrimp that we described a few years ago. So this also happens to be a blind groundwater dwelling shrimp. So they are fish and they are shrimps as well. We've not yet found any mollusc. So that's something that we would really be interested to look at in the future. So this one is also of interesting biogeographic or which has an interesting biogeography, because the other members of the family are all distributed in South America and Africa. So possibly this is an ancient relic dating back to the Gondwanan supercontinent. We don't know. So that's also a work in progress. So we also had some really good visibility for our work. So Hollywood superstar Leonardo DiCaprio talked about our work and how citizen science really contributes to the discovery and description and conservation of subterranean and often unexplored habitats and overlooked fish species. So this was indeed a great visibility for our work. All of this was covered around the world in different national and international media. So this was basically, as I said, an endorsement for the approach that we take, citizen science-based approach that we take for understanding and documenting groundwater fish and other fauna of southern India. So from a very poorly known group of fish, as I showed you in my introductory slides, based on a few black and white illustrations for the last 50 years, we have been able to improve the knowledge base on these fish from possibly less than 10, 20 records in the last 50 years to more than 500 records in the last 7, 8 years. We've described 8 new species. Some of them are also new genera and new families that I talked about. So basically, the common public has helped us a great deal in discovering and understanding these spectacular fauna that live under our feet in the southern part of India. Lots of media support for us. So the media has been very kind enough to cover all our work. So they even incentivize this in a way that they put out photographs of the villages and residents who have helped us discover new species. Give us lots of new information. Also, it's like a win-win situation for both the members of the local communities as well as us scientists trying to unravel these mysteries. We also used a lot of science, just not citizen science, but also hardcore science. We use advanced anatomical tools, CT scans, to understand the evolution, evolutionary morphology and things like that. We also use a lot of advanced molecular tools, for example. So we're standardizing environmental DNA technology for detection and monitoring of these fish. Obviously, we think that this is the way to go to understand fish from these inaccessible habitats. So just looking at the water and trying to find out the distribution of these species. So that's also something that we are developing right now. So the main question is, will these really ancient fish survive the sort of extreme impacts to the habitats that are occurring in southern India and also elsewhere in the country and obviously almost throughout South Asia. So this is a very depressing map. This shows that India is one of the regions which will face extreme freshwater shortage in the next 25 years. So it's not a prediction for 100 years or 200 years. So it's just a prediction for the next 25 years. So in 2050, it's predicted that almost 50 million or more than 50 million people in India would go without freshwater. And obviously you can think of what would happen to these interesting fish that live in these groundwater systems, because groundwater is one of the most important source of fresh water for everything that we can think of for human uses, for irrigation, agriculture, and so on. So that's a depressing sort of prediction we have. So the question is what do we do to ensure that these fish survive the Anthropocene? How do we secure a bright future for these fish? The threats, lots of unmanaged groundwater extraction that is happening throughout the country. The water table is going down on an annual basis in several parts of India. Still, lots of use for freshwater. So this is one of the biggest threats for these fish. There is a lot of reclamation of wetlands for development, apartments, shopping malls, different other developmental activities, happening throughout the country. So there's lots of reclamation of wetlands, reclamation of paddy fields, deterioration of wetlands that are still sort of not used for any of these activities. A very interesting identity crisis in India. So we've all heard about human animal conflicts. So we have an interesting case of human fish conflicts in this part of the world, where people mistake these interesting fish for worms, for snakes that are poisonous or toxic to the water. And so the first reaction for residents once they come into contact or once they collect a fish from these wells are to kill them, and of course think that the water has become completely poisonous or toxic. One of the reasons is that this region is also known for its waterborne diseases, especially during the monsoon, the rains. And so there is a tendency among health officials, health department officials in the country to disinfect and chlorinate all of the wells. And these fish are super sensitive to any small changes in the water quality. So, chlorine just kills all these fish. So we've been trying to educate and make the villagers aware that these fish are really ancient, 50, 100 million years of evolutionary history. And they would really be the first to sense or understand or detect any small changes in the water quality in the wells much before we humans do. So we are trying to advocate that these fish should be used rather as an indicator of good water quality than considering them as toxic or poisonous to the water. But again, this is something that we are struggling with because of the trade-offs between human health concerns and biodiversity, for example, because there's no alternative to chlorine-based disinfectants for well water. And when it comes to an argument of saving human lives against saving these obscure fish, it's always a bias towards human health related concerns. So that's was my last slide. Thank you very much. And if there are questions, I'll be happy to take them after all the talks.
Thank you so much, Rajeev. I always find that one of the things that's most striking about your work into subterranean species is how entwined it is with the local communities. Obviously, they're the people who live by the springs and experience them daily, and have access to them so much more than the scientists do generally. And it's just sort of wonderful to see how collaborative it is with the local communities. So thank you for that. And now to the diabolic springs of Death Valley. Oh, you're on mute, Chris.
There we go. Well, somebody had to do that.
It wouldn't be a
Okay.
webinar without someone doing it.
I know. Well, it was great hearing from all of you so far. Those have been some fantastic presentations. I particularly, Rajiv, love those photos of subterranean fishes, cave fishes. I've long loved that diversity. And Michael, thanks for bringing us all together today and focusing attention on spring systems and freshwater fish conservation and extinctions in general, right? I've long agreed that when we look at fish extinctions and fish conservation threats, it's almost entirely freshwater. It's not this sort of marine focus that we're often sort of the public is sort of more thinking about. So I'm the curator of fishes of ichthyology at UC Berkeley. We're the youngest scientific fish collection. I'm also associate professor there. And fishes are more than half of all vertebrate diversity, right? And we are fishes, right? Our ancestors had fins. All of these tetrapods and mammals and reptiles are nested within fishes. So there's so many things to sort of love and appreciate about fishes and fish diversity. This is what one of our ancestors looked like in our family tree. At the same time, we talk about conservation, and all of these species have this right to exist, and they have their unique evolutionary stories to tell. But part of that story can also inform us about human health. And so, I mean, Rajiv said there certainly is this sort of trade-off between conservation and human access to water rights, but it's also worth remembering when we lose these species, we lose the ability to look for biomedical insights into some of these species. So my own lab has been funded by the National Institutes of Health for the last almost 8 years. My postdoc, Fania Palomenos, who's in the audience, has also had funding to work on endangered pupfish species in order to learn about human health. My lab has focused on these sort of desert-like environments, these hypersaline lakes in the Bahamas, Devil's Hole in the middle here, which I'll talk about mostly today, as well as these isolated crater lakes in Cameroon, where we get just, rare rapid evolution of new species because there's few predators. And this is sort of the big elephant in the room, right? When we think about biodiversity, it really is these remote, isolated habitats that are more vulnerable to extinction threats because they are so isolated. And that also leads to rapid evolution of new species. My particular focus, as Michi sort of first introduced here, are pupfish, which are called puppies, named after puppy dogs because they like to play like puppies. So their range here is shown in blue, as well as all the way into desert regions of the US and Mexican deserts, as well as these sort of coastal areas here in blue. My lab has often looked at the rapid evolution of new species in two different places, which we still don't really fully understand why. But San Salvador Island and Laguna Chichen-Canab, where we get rapid evolution of trophic specialists in hypersaline lakes like these in the middle of this island. So here is one of the only places in the whole range of pupfish where you get 3 or 4 species actually coexisting in exactly the same habitats in the same lakes. And how are they doing it? They're specialists. So one of these is a scale-eating specialized species that bites the scales off of its prey. In Laguna Chichen-Canab, it's a really similar environment. There's absence of predators. And we went there in 2022 with collaborators. We found all 3 species still there out of the original 5. So this radiation had already known to be, threatened by invasive species that were introduced into the lake in the 1990s for aquaculture. And we particularly found this one with big lips called labiosus, as well as the zooplanktophore species called Simus. So this was in 2022. We were like, great, I can keep working on the system the rest of my career, right? We also found a new invasive species, which is actually a native species to the Yucatan, but it had never been found in this lake before. So the lake level was rising, potentially reducing the salinity of the lake, allowing a native species to colonize in around 2019, 2020. We go there in 2022, find all 3 species. I return with a massive team of collaborators from local students, students from Mexico City, 2 years later. So intensive fishing efforts across the entire lake. You see where this is going, right? We found 1 of the 3 species, and we found lots more of this native Mayan cichlid, and its stomach were full of pupfish, right? And most of these are the invasive cichlid. So extinction is happening extremely fast out there, right? This is not reported in the literature yet. We've done more expeditions. We still haven't found those native species. So that zooplanktophore is extinct in the wild. It's only held now at the London Zoo and the Vienna Zoo. And that thick-lipped specialist, even though, 10 years ago I had it in my lab, other people had it in Spain, it was all over in situ conservation, is fully extinct as far as we can tell. There's no captive populations. And this is true for 3 other species of pupfish that are extinct in the wild, only held in captivity. There's, and then as many of you know, there's efforts through the London Zoo and others to try to census these populations as well as reintroduce them to the wild. But what I'll focus on today is the devil's hole pupfish. So this is probably arguably the most famous desert fish in the world, right? It has one of the smallest habitat ranges of any vertebrate, I would argue. I think that minnow might be a contender too, Michi, but the entire habitat of this pupfish is behind me there in this photograph. It's a little blue fish, it's about a couple centimeters long. It's nested within this sort of extremely beautiful desert landscape of Death Valley in Nevada. So this is another creek that runs through Death Valley that's a year-round water source called Salt Creek that also has an endemic pupfish species, but one that has never really been threatened. The area has been protected since the 1950s. This fish has been doing just fine. Whereas devil's hole pupfish is extremely actively managed. So you can see to the right of me here is all this equipment that's permanently in devil's hole. They monitor the water levels. They actually feed the fish every day with automatic feeders. There's 5 full-time staff at least that their entire job is dedicated to conservation of this fish and funded through the U.S. Endangered Species Act. And this species, back in sort of the early 70s, protection of the species went all the way to the U.S. Supreme Court case, where the pupfish actually won the case. And that case is still taught in all law schools in the U.S. as a fundamental case affirming federal government's rights to groundwater underlying Devil's Hole. Devil's Hole actually goes down at least 100 meters or more. No one has ever found the bottom. It's now razor wired off because it used to be a popular cave diving site. It actually opens up underneath there into a massive underground cavern with sort of extra exits. And the pupfish actually only go down about 30 meters, and they mostly live and breed on this little shelf right here next to me. And then about 2012, there's been other refuges in the past. But in 2012, they had some money within the park system to actually build a second replica Devil's Hole. So this is a 100,000-gallon pond about 1 kilometer down the road, which uses the same groundwater pumped into this system. There's a crane in there. They actually did a laser scan of the shelf and then recast it with resin to try to mimic this shelf right here. So you can sort of see the resemblance right here. Here's some of the staff that have worked here. All of these folks have now moved on. None of this is actually open to the public. We only were able to go through the Desert Fishes Council organization. And now there's about 500 pupfish that are living in this second refuge population for Devil's Hole, of Devil's Hole pupfish. Whereas in the wild, it actually dropped down to just 20 individuals this past January. So 20 in the wild, over 500 in the refuge. It's sort of a resounding success for that refuge. But this whole effort took almost a decade of sort of building it, gradually introducing fish. They couldn't find the fish when they introduced them, sort of slowly recognizing how do they actually manage this refuge. It got colonized by predatory diving beetles, which would eat all the eggs. And so they had a full-time staff person hired just to kill diving beetles for about 1 year. And that turned out to be the solution. So I could go on and on, right? There's so many stories about this fish. I should add the population is actually counted twice a year, right? So there's multiple divers that go down all the way since the 1970s. So we have a really accurate census population size of Devil's Hole pupfish. And it has always sort of fluctuated between about 100 individuals up to 500 individuals in the fall. In the winter season, for 3 months of the year, Devil's Hole is actually so far down in that cavern that it doesn't even receive direct sunlight at all. And this is a fish that mainly eats algae, right? So their entire food source basically disappears for 3 months of the year. They're basically just starving, and their population does crash during the winter. And then, as you notice, around the 1990s, mid-1990s, that population started to crash overall. We still don't know why. Going down to population lows of just 35 and 37 individuals. Some of that was actually management human error where they left a trap in there. And then going all the way to this year, which we don't have on this graph yet, but the smallest population ever recorded was due to multiple earthquakes in Mexico back to back, which underground are connected to Devil's Hole and actually cause tsunamis within Devil's Hole, which then scrub that algae shelf clean of algae. So around January, the fish are already starving, and suddenly back-to-back tsunamis further dump rocks in and cover all of their food source. And I think those 20 probably only survived because of the supplemental feeding from that feeder, which dumps in some pellets every single day. What I'll just talk about sort of relatively briefly today is my grad student, David Tian, just graduated. He's now an MSF postdoctoral fellow at UCLA, but he did his whole dissertation on conservation genomics of the Devil's Hole pupfish. So it took a long time, almost a decade of me asking for tissues to finally get tissues. We actually they had one aging elderly Devil's Hole pupfish in captivity that they euthanized weeks before it was going to die so that we could do a new de novo genome for this species, which was, that was hours and hours of talk. But essentially only one pupfish was euthanized for this study. Everything else were fin clips that were collected from refuge fish that had been they collect the eggs from the wild, they raise them in the refuge, and then those wild collected eggs are then introduced to the refuge. So none of this is impacting the wild population at all. But long story short, we sequenced about 50 whole genomes of Devil's Hole pupfish, as well as de novo genome assemblies. And we were actually able to go back to museum specimens all the way back to 1941. So formalin-fixed museum specimens, and sequence whole genomes of these as well, using this sort of museum DNA extraction protocols that are working surprisingly well. So some of these fish were getting, 5x coverage of the entire genome. And so this has basically, long story short, right, this has the lowest genetic diversity of basically any wild vertebrate population that's ever been measured. Devil's Hole pupfish here is all the way at the bottom. There's an island fox population that has equivalent low levels of genetic diversity. And to put that in context, here's another Ash Meadows desert pupfish in the neighboring region. This pie is just an estimate of genetic diversity across the genome here. So this is Ash Meadows pupfish. This is Devil's Hole pupfish. So across a billion base pair genome, we're seeing only about 60,000 sites where there's any variation at all. So extremely low genetic diversity. And we find that even in those historical samples going back to the early 1940s, that genetic diversity is just as low back then as it was in about 2022. So previous to the sort of the recent bottleneck, but post the sort of first bottlenecks that were human caused, right? And so that's sort of good news, is that most of the human management of this population has not impacted genetic diversity. They've always had extremely low genetic diversity. And at first we were like, oh, this sounds bad, right? But this kind of makes sense, is that refuge population, which now has 500 fish, has about half the genetic diversity of the wild population. And this kind of makes sense, right? And even though actually every year they put carpets out into the wild Devil's Hole for about a week, and they pull the carpets and then they search through the carpets where some of the pupfish will lay their eggs, and they pick out each egg and raise it in a little petri dish. But that's still a subset of that wild genetic diversity that they're introducing to the refuge. So even though they're introducing fish every single year, they're still not fully sampling all the genetic diversity that's in the wild. And further, you add to that is that pupfish are lek breeding, very territorial, aggressive fish. So one male in that refuge population can dominate all the matings, or most of the matings. And that also is going to reduce genetic diversity both in the wild and the refuge. What we found, though, is that also these captive refuge fish are more inbred, right? So they're more likely to result from recent sort of, aunt-nephew sort of matings within their recent history. And so this red regions are regions of the genome where we see no genetic diversity, that seem to long regions of just no diversity at all that seem to result from inbreeding. And then the interesting thing here, which I recommend as sort of a management consideration going forward, right, is that this is so intensively managed, and each egg is raised in the refuge, that the managers started to notice, wow, some of these eggs started to develop about 5 days, and then they died, and their heart rate slowed down. And we realized that if we just measure inbreeding of all these eggs that died that we also sequenced, compared to adult fish from the refuge and the wild, is that if you're more inbred, it actually predicts how likely that egg is to die during development, right? So even at the most extreme levels of low genetic diversity still matters, right? Even in this refuge population, which we were quite surprised by. This is what that looks like. So here you can see the heartbeat of the wild embryos, as well as, so both of these eggs are collected from the wild, from those carpets. They're raised in the lab, and on the right, you can see the melanocytes have expanded more, and the heart rate is actually slightly slower, which we measured from essentially iPhone videos. And then with these slowed down dead embryos, we actually do think we found one region of the genome that is associated with death in about 14 out of those 20 embryos that we sequenced that died prematurely. And that region contains one single variant that all of the dead embryos have, that's an amino acid change within a heart defect gene, right? So we think of that as kind of a smoking gun. We may have found this region that's actually killing those embryos. So we want to test that more in the lab for sure, right? But also, when we look at all these genomes, it's an extremely powerful data set. And as you might expect, we actually find 37 genes that are in the Devil's Hole pupfish that are just completely broken. And comparing that to neighboring desert pupfishes, all of them have functional copies of this gene. Devil's Hole pupfish, they're broken, right? Which is probably a big reason of, resulting from inbreeding. This is why they're so hard to breed in captivity. And we even find regions of the genes that are just completely missing. So deletions of genes that are not detected in other desert pupfish. One of these is really interesting, right? Because it's related to DNA repair. And we see a deletion in this. Does that result in mutation rate? DNA repair, of course, is relevant to cancer in our own genomes. And so further studying these can also be relevant for human health. So lastly, I want to point out, right, this was 2024 in May, sort of a look at how awesome we are article in the New York Times. The resilient pupfish makes a comeback. And then we have April 15th, 2025, earthquake nearly wipes out the Devil's Hole pupfish. Colony drops to 38. I don't even have the most latest article when that 38 is actually 20. So that additional number is they took refuge fish and dumped them in the wild and then issued a press release to make it sound a little better than it actually was. But that shows just in the span of 1 year, right? You think you're having success, but these species are extremely fragile, right? And so we always have to sort of keep our guard up and have those refuge populations. Like situ conservation surely saved the species in this case. So future directions, where are we going with all this? I feel like at the genomic scale, I feel like we've done a lot, right? We already have a good idea of what genes are broken, what needs to be fixed. With fish systems like this, it's actually relatively easy to use CRISPR. We can fix these broken genes, restore them to historical genetic diversity of the 1930s and 40s in the lab. If you think that's wild, this has already been done for white-footed ferret in some ways, where they cloned a ferret from museum skins from the 1980s. I mean, sorry, the 1880s, right, in a modern ferret so that they could reintroduce genetic diversity from the past into the modern population. And my lab has got CRISPR working in pupfish. This is one of our Caribbean pupfish where we knock out a pigmentation gene. And you can see this pale phenotype, which we've now verified. We've introduced mutations right where they should be. I have undergrads in my lab doing this. This is an incredibly cheap technique now to break genes and test their function, right? And with that, I want to leave plenty of time for discussion of the panel as well. But just in summary, why is genetic diversity so important? This is the least diverse population in the world, but even so, inbreeding strongly predicts embryo survival. That might be sort of genome-wide inbreeding, but we also think we have one particular candidate locus that seems to predict embryo survival across the board. And we've identified lots more candidate mutations that might be reducing the overall health, genetic health of devil's hole pupfish because of its long history of isolation. There's so many other stories here that I haven't talked about, right? The top predator in Lake Titicaca was a pupfish, now extinct, only held in about 20 museum records worldwide. No photograph to even show you. This is the outgroup to all pupfishes, the most close related species, Megupsilon. I saw the last male of this species go extinct in my lab. The last female went extinct behind the scenes at the Dallas Children's Aquarium. So species extinctions, especially of freshwater fishes and other freshwater groups, is happening so rapidly and not getting the attention that it deserves. So I'm really happy to be part of this workshop today and panel today. And with that, I want to sort of thank my lab, particularly David, the work I talked about today, my postdoc, Fania Palominos, who pioneered the CRISPR approaches in my lab, and lots of funding agencies. And thanks for your attention.
Amazing. Thank you so much, Chris. That was a really fascinating, rattled through a lot of information there. Really interesting. So just a quick one from me. With the Devil's Hole pupfish dropping to just 20 individuals over the past few months, which is an astounding number, what have been some of the implications for the management for your research?
Sure. Yeah, thanks for that question. It is kind of fascinating where we had told the managers, hey, the refuge population has half the genetic diversity of the wild, right? And they knew that. Then this actually crash happened about 2 weeks into the Trump administration taking over and just firing fish and wildlife managers left and right. And they were kind of huddled in a corner, dark part of their office, like we're going to be fired any minute. And apparently they took 10 days to make this decision. And after 10 days, they didn't consult any scientists outside of the group. They told my lab not to come. We were planning a trip. They were like, don't come. We're busy. We can't tell you why right now. And then they took refuge fish, which they knew had half the genetic diversity, and they dumped them into the wild. Refuge water and all, that just dumped them in. They didn't take any fin clips. There was recently an article in National Public Radio for the US, right? Sort of where I'm criticizing them a little bit for this, right? Where we get stressed about conservation, and there's always this sort of balance between the science and the conservation, and what do you do? And, at this point, because they introduced those refuge fish with no genetic samples at all, we have no idea now what the wild population is. Is it entirely refuge fish? Probably, because those refuge fish were extremely fat, extremely large, extremely happy fish that grew up with plenty of food, and those males and females now dominate the matings. And they even report this. They're like, oh, wow, we saw fry right away. Well, there shouldn't be fry that early in February right? And so what happens is those refuge fish go out there, they dominate the matings. But in the long run, we know that the refuge fish have more of these lethal alleles that are basically genetic variants that are causing those embryos to die. Those are now dominating the wild population as well, right? And so just always taking into account the genetic health of the population and that the wild populations are still always going to have more genetic diversity than our refuge populations. It's just a really delicate balance that needs a lot of consideration and forethought, I would say.
Yeah, okay. Thank you. And there's there's a couple of I see that Julie has put a link in the chat. Devil's Hole, pupfish, crisis federal science, Trump. I've not clicked on it myself, but I assume that's exactly what it sounds like due to policy decisions there. There's a new crisis or an updated crisis for the pupfish. Um, Izzuddin Al Faruqi, I hope I'm pronouncing your name correctly, has said in the Q&A, Izzuddin is an ornamental fisherman from Kalimantan in Indonesia, and he's actively searching for fish from East, West, and North Kalimantan. And he has three questions to the entire panel. So if anybody has any suggestions or answers, please jump in. Is it possible for them to work with citizen scientists? They're exploring an upper stream in a small river upper stream in Kalimantan, looking for some ornamental fish, and they're keen to find collaborations to discuss next steps. One of their biggest fears is if they share the location, people will go there to catch the fish. It's a very valid fear. Is it possible to get to work with collaborators? And if so, how? I don't know if anyone has any suggestions for that. If not, Izzuddin, I suggest you if you write to info@shoal.org, then we'll get back to you with a fleshed out answer. He
There is,
also asks, oh,
Michael, there is an Indonesian species specialist group based in Indonesia, and there's the Pili Green network that are both active working in terms of, at the moment, the ministry, I can't remember if it's the Ministry of Wildlife or the Ministry of Forestry, have an active red listing program going on there with the intention of doing quite a few species by the end of the year. But the person to get in touch with would be the Indonesian species specialist group. And there is a program officer Celia, whose name escapes me, but I'll pass that through to you later. The only caution I would have is that movement of fish around has quite a big impact on the other species in the lakes and rivers there, and quite a lot of the crustaceans and molluscs are basically on the edge of extinction as a result of fish being moved around. So I don't know, I can't recall off the top of my head whether those are native species that have been moved from where they ought to be to somewhere where they're not, or whether those are invasive species. Yeah, I think Ikram has just picked up a point. He's somebody who was at our mollusk workshop. He's answered in the webinar chat.
yeah, brilliant.
Points out that eDNA would be a way of finding that fish if you've got.
Yeah, that's a good point. And do you maybe have a contact for the Indonesian
I'll
specialist?
post it. I'll post it for you.
Yeah, if you have it there, that'd be great. If not, Izzuddin, please write into Shoal and we'll get back to you once we have that. And I think that actually answers your second and third question as well, which is about how do they know what they're doing is sustainable for the environment. And the biggest problem during catching the fish is insecticides. 2 or 3 people can kill the entire river fish population. How can they stop local people from doing this? So Izzuddin, please, yeah, write into Shoal and we'll get back to you on that.
Another interesting concept might be Project Piaba, which is sort of spearheading sustainable ornamental fisheries in South America, and so they might have, valuable insights as to how to work with local fishermen to use these resources in a sustainable way.
Okay, brilliant. Thank you, Michi. We have 5 minutes left. I think that's probably time for maybe. Oh, Mary, thank you for that. So the contact is now in the chat. There's probably time for one question. Which one has anybody else got any questions from the audience? If not, Michi, perhaps you could say a little bit more about the ex situ conservation work and how it relates to Springs fishes, and if there is any formal conservation programs in place for the fishes that you were discussing.
Yeah, so the ex situ conservation motion passed along with the spring motion, and it has been sort of pushed forward in parallel, in part worked through Shoal, right? And there will be a workshop next week, actually, in Albuquerque, to sort of generate a global priority list of species that are in need of ex situ conservation, and in the hope to sort of motivate different stakeholders to start taking in species, sort of hopefully in the order of priority. And sort of the second part of this is to start developing best practices, so we're taking care of these species in a manner that they're sort of fit to go back, right? Chris mentioned this. These captive populations are valuable for conservation, but they're not without caveat. And you know, especially for small-bodied species that are not identifiable individually, we don't really have solid group management practices where we understand how do we assemble groups, how do we manage groups within an institution, but also across institutions, to maintain as much genetic variation as possible, and to prevent sort of adaptation to these artificial environments, to prevent domestication, right? And so that the hope of this group is to really push the envelope here, just like this group is pushing the envelope on spring conservation. This is relevant for spring fishes simply because a large number of the high priority species will be spring species. While there's efforts for a handful of species that are primarily species covered under the Endangered Species Act in the United States, where there's a federal mandate to take care of spring species, in most parts of the world, there is no effective conservation program, not ex situ or in situ. So there's a lot of work to be done, but there's also momentum growing. So.
Brilliant. Thank you so much. And Mary, yeah, very quickly,
Yeah.
we have to.
Just very quickly, I have to totally agree that one of the things that we need to be working on is good management practice of ex situ populations, and when you can reintroduce and when you shouldn't reintroduce, and at what point and how. And I think that also leads me on to one of the first things that we did was actually get a multi-species action plan together for the European freshwater bivalves. Usually action plans tend to be single species based, but it occurs to me that in this particular case, actually coming up with something that is multiple species across different taxon groups, so not just the fish, but maybe some of the things that are all together. So maybe actually finding a place where putting together a document that would be a conservation action plan that is multiple taxon and multiple different groups, and maybe fish, molluscs, dragonflies, crustacean, would be something that could then be rolled out as a model of how you go about developing it and how you can move it forward, because it's something we could probably get some funding for. And the conservation planning specialist group are really good at helping us do that. And I think we could bring in the reintroductions group. It's something that SSC, you could push to them and say, is this something that we could work on as a model that then could be rolled out elsewhere? We've just taken the freshwater bivalve model and tried it out in Indonesia, because we were dealing with a data-rich area in Europe, and then tried it out in a data-poor area. It was only last week, so I haven't had the feedback from Xander yet as to how that works. But Indonesia is certainly an area where it's a good place to actually test stuff out. So try something in the States, Australia, India, and then move it somewhere else. Sorry, I'll put my hand down now.
No, that's great. Thank you. Thank you for that, Mary. These sessions always fly by as far as I'm concerned. But unfortunately, we do have to wrap now. But thank you so much, Michi, Mary, Rajeev, and Chris. We really appreciate you taking time out of your busy days, busy schedules to speak with us. It's been really fascinating to learn more about the cutting edge research into springs and hear about some of the remarkable biodiversity found in them. If anybody in the audience has any questions that weren't answered during the webinar that you didn't think to ask during the webinar, perhaps, please email them into info@shoal.org and I'll forward them on to the panelists and get back to you very soon. As mentioned, this webinar has been recorded, so from tomorrow it will be available on YouTube. Feel free to share over social media. And if you'd like to rewatch, or know of anybody who was hoping to watch but couldn't make it, you'll find it online from tomorrow at youtube.com/shoal_org, or go to the Shoal website, shoal.org, news sections, videos. And also it's worth mentioning that Chuli Yu, Shoal's Chuli, has written a post about springs for the Shoal blog, which is really interesting. If you've not taken a read of that yet, please do head over and take a look at that. We have one more Season on Springs webinar, which will take place on the 25th of June at the same time, 3 till 5 BST. And you can find the sign-up details over at the Instagram, LinkedIn, Facebook pages. We'll also include them in tomorrow's newsletter. If you're not already signed up to that, please do head over to the website and put in your email address. It'll really help keep you updated with all of Shoal's news and the wider partnership, and also the wider world of freshwater conservation more broadly. The third and final session next month will be all about the conservation and policy for springs, which is a subject thankfully now gaining momentum, in part due to Motion 16 being formally adopted by IUCN at the last year's World Conservation Congress. So make a note in your calendars. It promises to round off the season nicely. Thank you all so much for joining us. And thanks again to the panelists. I hope you enjoy the rest of your day and hope to see you next time.